Well, so the time is up. Good afternoon, everyone. So welcome to our sedimentary forum, number 23. Today, we are honored to have invited David Haig to give us a talk. Before this talk, please allow me to give a brief introduction. David has expertise on Foraminifera, having the experience of teaching at the University of Queensland and the University of Papua New Guinea. He joined the University of Western Australia in 1985 and retired in 2010. He is now an active senior honorary research fellow in the Oceans Graduate School at the University of Western Australia, and is an honorary stratigraphic advisor to the Instituto do Petróleo Geologia Instituto Plateau in Timor-Leste. David was head of the Department of Geology and Geophysics from 1995 to 1997, and deputy head of the School of Earth and Geographical Environmental Sciences during Lynn Albert's leadership in the 2000s. He joined the Oceans Graduate School in 2019 because his paleoenvironmental research aligns with some of the work being done by Malcolm McCulloch's research group, and with other work on marine environments within the school. Because of his outstanding and substantial contributions to the geoscience in Western Australia, the Geological Society of Western Australia branch awarded him its Gabe Maitland Medal in 2018. David is an explorer of lost seas and their modern analogs. His research focused on the development of the Indian Ocean from precursor interior continental seas through a passive margin and then to the closing of the northwestern part of the ocean via collision of the Australian and Eurasian continents. He studies changes in marine environments, climate, and biota over the last 350 million years within a tectonic stratigraphic framework. Foraminifera offer unique solutions to many scientific issues in both geoscience and biological environmental sciences. In this talk, David will present 4 aspects of research based on an overview of 50 years research on Foraminifera. So, David, please can you start your talk now?
Yes. Greetings to everyone from Perth, Australia. And I sympathise those of you in COVID lockdown and with COVID. I had COVID 6 weeks ago, so I know what it's like. And thank you, John Wu, for inviting me to give a talk specifically on Foraminifera. I have studied Foraminifera for over 50 years, and in that time I've travelled back to the Carboniferous and through the Permian, Triassic, Jurassic, Cretaceous, where I started my career, Paleogene, Neogene, and into modern seas. From the overview of this work, I want to provide some thoughts about the main directions for future foraminiferal research, the sorts of studies that I consider to have future relevance and that I would like to continue if I could live another 50 years. Today, our science is undergoing great change, not only in terms of career opportunities, but also in greater access to exciting, new, high-level technology. So in this talk, we'll go back to the future. Foraminifera are incredible single-celled organisms. Look at how beautiful this test is. Can you see the arrow there?
No, I can't see the arrow.
No, I just
Still
put that
in
on.
the first page. Yeah. Can you
Yeah.
put in your slideshow? Ah, yeah, I can see the arrow now. Yeah. Yep.
Right.
Yeah.
So this test is of Paneropolis planatus, and it's from Shark Bay, Western Australia. We caught it reproducing as it lived on a seagrass leaf. Seagrass leaves like this, and some of these white specks are Paneropolis. It built an agglutinated cyst around the terminal face of the last chamber, which has apertures to the exterior. The protoplasm of the parent that built this shell has subdivided to produce many embryos. And here you can see some within the shell that haven't escaped yet, some that are escaping, and others that have escaped and make their way out into the wider world. And you can see that the embryos have acquired a portion of the unicellular algal symbionts that lived in the protoplasm of the parent and give a purplish colour to the living foram. The symbionts provide additional oxygen and additional nutrients to allow these minute embryos, less than 100 microns in size, to build a shell like this over the period of a year that reaches 1.5 millimetres in size. Over the years, my work has looked mainly at the geological history of the western and northern margins of the Australian continent, from an interior rift in eastern Gondwana to opening of the Indian Ocean and leaving the passive continental margin and these marginal continental plateaus, and then to the collision on the northern margin of the continent. And in this talk, I'll focus on 4 areas that I think I would like to continue future research on if I live another 50 years. And these are living forams, molecular DNA studies, high-resolution environmental change studies, and reconstruction of tectonic histories. So let's look first of all at the living forams. And of course, forams are living organisms, and most end up when they die as sand grains on the sea floor. And we have to remember that the living populations are not equivalent to post-death assemblages in sand or to fossil assemblages. For example, species have different reproduction rates, so altering the proportions. And seafloor sand is mixed by bioturbation and various sedimentary processes. But knowledge of living forams should inform all foraminiferal studies, whether it be in the late Paleozoic or in the Cenozoic. I'll give examples of 2 small studies that we have recently undertaken, one on carbonate platform facies and the other on basinal mud facies in an estuary. Now let's look at, anyone can go out to the seaside and look at living forams. They're everywhere in marine environments and estuarine environments. So this is on a carbonate bank facies. This study dealt with the miliolid Vertebralina striata, living as an epiphyte on the seagrass Posidonia in very shallow water on a modern carbonate platform in the Perth region near where I live. This study was led by one of my students, Clement Tremelin, and has just been published. Here is a typical Vertebralina striata with very low trochospiral tests that you can see on the spiral side. On the umbilical side, there's an aperture on the underside of the last chamber. So this form lives, as you see here, with the umbilical side down, the aperture down on the seagrass leaf. And it mows up, it is mowing up food on the leaf surface, for example, diatoms and small fragments of organic debris. You will notice that the seagrass here, over here, is covered in filamentous algal epiphytes. This is Phacelia, the algae, the filamentous algae. These occur on mature leaves, particularly at the end of summer, before they are shed to form part of the seagrass rack that often accumulates on beaches during winter. And Vertebralina lives among the Phacelia and the other epiphytes on the leaf surface. Also notice these Vertebralina, which were picked from among the Phacelia. They have bizarre divisions of the final chambers. You would perhaps think, especially if you saw these in thin sections of limestone, that they were a different species or even a different genus from these. Down here on the lower right, these images show how Vertebralina builds a new chamber. First, a flexible gelatinous sheath forms the outline of the new chamber. And then minute calcite crystallites concentrate in certain areas beneath the sheath, forming these white patches. Note that the ornament, you can just see it here, is already impressed on the sheath before mineralization of the wall. What Clement in the study realized was when Vertebralina was trapped, like this one, between Phacelia filaments, these filaments formed an impediment for normal chamber growth, as shown here, the blue of the gelatinous sheath. This flexible sheath allowed the chambers to subdivide around the filaments, as shown here. This results in these types of tests. So in studies published elsewhere, these abnormalities in Vertebralina that were collected as dead specimens in marine sand have been attributed to chemical pollution. This is not correct. They are the product of competition for space among epiphytes on the seagrass leaf. So it's important to know how forams live and the microenvironments in which they live. Now we turn our attention to mud facies faunas. And these examples are from a living assemblage of Ammobaculites here in an estuary, these are from the Wellstead estuary, southern Western Australia, with seasonal high freshwater influx. Estuaries are really just small versions of larger, shallow water interior continental seas. Now, these are agglutinated forams. Their agglutinated Foraminifera are usually incorrectly lumped together. And I want to emphasize this. They are represented in 4 separate, unrelated Foraminiferal classes. The first one, like Ammobaculites here, is an organic cemented, non-calcareous agglutinated class. There's no calcareous material incorporated in the wall here, and the grains are held by organic cement. The second class is certain groups of Phylogenata. The third class, certain species of Miliolata. And the fourth, the carbonate cemented agglutinated types, which have more affinities to the Phylogenata than to the organic cemented agglutinated types. Note in the estuaries, only organic cemented siliceous agglutinated forams occur here. Out in the open sea, you get carbonate cemented agglutinated forams, which are quite different to these ones. Ammobaculites has an abundant fossil record in mud facies of shallow water interior seas and estuaries, ranging from at least the Carboniferous to the present. Some Paleozoic workers who are not familiar with living faunas or their environments think that these, and this is from the Permian, Triassic, Cretaceous, think that these are silicified calcareous types. And that is nonsense. Hypersaline estuaries, or those with seasonal high freshwater influx, lack carbonate cemented agglutinated foraminifera and only have the organic cemented siliceous types amongst the agglutinated fauna. They, of course, also have calcareous forams without agglutinated walls in their grains, mainly rotolines such as ammonia and Ophidium. I recognised this very early in my studies of the Cretaceous, so the great interior marine basin in Australia, this huge Cretaceous interior basin. The mud-dominated interior sea had only organic cemented, this OC, agglutinated forams among the agglutinated assemblage, including Ammobaculites, these ones down here. I call this the Ammobaculites Association. Nodosaurids and galvalinellids that are calcareous but non-agglutinated are common in the Ammobaculites Association. The open marine continental margin assemblages had both carbonate cemented, CC, and organic cemented agglutinated types, and diverse other calcareous forams. Organic cemented agglutinated forams, which mainly agglutinate silicate minerals with great specificity in grain size and mineral composition for different species, belong to different genera, different families, orders, etc., to the carbonate cemented types. The ability of the foraminiferal shell to precipitate carbonate cement was a fundamental step in the evolution of Foraminifera. Now let's go on to talk about molecular phylogenetics studies influencing taxonomy. And I want to deal with microevolution. By that I mean species to species affinities. And then macroevolution, the taxonomic scheme, really a phylogenetic scheme at generic and suprageneric levels. So the species to species affinities. Over here, you can see a molecular phylogenetic tree for 51 living agglutinated species. Note that it's based on comparison of a selected fragment of an RNA gene, a fragment of the gene. The tree is rooted in the uniserial organic cemented agglutinated genus Rheophax, which first appears in the fossil record in the Ordovician. It's the first foraminiferal genus to acquire a multi-chambered test. The 51 species here that Maria Holzman and her colleagues analysed include both organic cemented as well as these carbonate cemented types. Note that the carbonate cemented types cluster together. They're not related to the organic cemented types, in my opinion. Note this cluster on top. This is identified as Trochamina hadai. formerly found originally in very shallow seas in Japan, then in San Francisco, over here, where Mary McCann considered it invasive. In our recent work, again led by Clement Tremlin, in this coastal inlet in Western Australia, we found Trochamina, these forms here, living at the surface of a flocculent mud layer in about 4 to 5 meters of water. Here you can see it living specimens. In terms of morphology, it appeared to be Trochamina haegi. And Maria Holzmann in Geneva confirmed this through molecular comparison. In Japan, Trochamina haegi is common in estuaries where there are wood chip processing factories. And this port in Western Australia exports wood chip to Japan. We think that Trochamina haegi has come into this inlet in ballast water from wood chip ships. No other estuaries in Australia have this species. So molecular comparisons are a definitive way to confirm species identifications that are suspected from morphological comparisons. Now let's turn our attention to macroevolution, the suprapacific classification. You'll notice that in the molecular phylogenetic tree, the Trochamina haegi cluster is very remote from the type species of Trochamina inflata, which has been recorded worldwide in estuarine marshes. Trochamina inflata lives close to the Rheophax root of the tree. Now, it makes sense that a Trochamina morphotype should be among the first of the organic cemented agglutinated multi-chamber foraminiferal groups to branch from uniserial multi-chambered Rheophax in this molecular phylogenetic scheme. Trochamina is the earliest foram of any foraminiferal group to develop a trochospiral test. And the oldest known Trochamina was found in the Mississippian shales in the US, where it's poorly preserved. We have found almost perfectly preserved tests in the earliest Permian mudstone in Western Australia. And Clement Tremlin has compared these to their close living morphological analogue, Trochamina haegi. But Trochamina haegi here is remote from the supposed primitive Trochamina inflata. This is Trochamina inflata. And of course, the Rheophax rootstock. And if you look at Trochamina inflata, you can see it has a polished wall. These early forms here have a rough wall exactly like Trochamina haegi. And we think that Trochamina inflata is a derived form, a very recent form in evolution of Trochaminates. So this scheme doesn't make sense. Now here, note that the oldest calcareous trochospiral forams are in the early Triassic, or perhaps in the latest Permian. And you can see that these organic cemented forms were developed prior to these. So clusters seem to be out of place on this molecular phylogenetic tree. Now, I believe that you have to consider molecular phylogenies very carefully and make sure that they follow or add to well-documented stratigraphic phylogenies. This is the high-end classification in the worms taxonomic database that is put together by Bruce Hayward and others, that is partly influenced by recent molecular phylogenies. And this classification of the class Nodosariata makes no sense at all. In particular, the inclusion of organic cemented agglutinated taxa, Rheophax, in the same class as Nodosaria, which has a radial fibrous calcitic wall. You can trace the evolution of the Nodosarians from down here in the, these are specimens from our early Permian, but they start down here in the mid-Carboniferous. You can, their forms, the earliest forms, are forms with a proloculus followed by a tubular second chamber with a simple rounded aperture. Next forms are forms with constrictions in the chamber cavity. And then in the early Permian, latest Khazalian or early Permian, you have forms with retain the simple aperture, rounded aperture, that develop true septa between chambers. Then in the Artinskian, you can see the inception of a radiate aperture. And in the dentalinids, up here in the early Triassic, you have the inception of costate ridge-like ornament on the shells. Now, these are forms. I'm revising the classification of the Nodosarians, both in the Permian and in the Triassic. These are dentalinids from the basin mud facies of Timor. For Nodosarian forams, you cannot devise species diagnosis and classification schemes from random sections in limestone. This is because you cannot determine in any one section the three-dimensional architecture of chambers or of the overall test, the nature of the aperture and lips or necks around the aperture, and the full extent of the ornament. Serial acetate peels or nano-CT scans may allow this, but not ordinary thin sections. I believe for stratigraphic phylogenies of benthic forams, you have to start at the base, the stratigraphic base, and work upwards. This is what I'm doing at present with the Permian and Triassic Nodosarians from the Eastcon 1 and interior rift basins. And I'm revising each of these Triassic families. The classification will be quite different to the classification in the worms database. There is something else important here for those of you who study mass extinctions. When looked from afar, there are very few cases of Nodosarian extinctions at mass extinction boundaries. For certain groups, the pattern you see above a major extinction level is diversification. The genus Lavidentalina, which first appears rarely in the Permian, diversifies markedly just above the Permian-Triassic boundary. And similar morphotypes crossed the Triassic-Jurassic boundary and continued through the Cretaceous and beyond. The genus Dentalina, which first appeared rarely in the Triassic, diversified substantially just after the Triassic-Jurassic boundary. Now let's come to high resolution environmental change. The bowl of rice rationale for many foraminiferal studies in the past was age determinations for petroleum companies and government geological mapping agents. I think that these days of being service providers to these agencies and companies are passing. One area that I think has been neglected over the last 20, 30 years has been the use of Foraminifera to plot out and help resolve the high-resolution environmental changes that we see in our sedimentary successions. And I'm talking about very detailed studies of Foraminifera, not just taking a species here or there. During the last 20 to 30 years, there has been the rise of sequence stratigraphy and chemostratigraphy in sedimentary basin studies. And detailed foraminiferal studies have not kept pace and have rarely been integrated into a sequence stratigraphic framework. I'll give two quick examples from our work in Western Australian basins to show what resolution Foraminifera can offer. One in siliciclastic mud facies and the other in carbonate facies. Over here are depositional cycles in the late early Permian in a shallow water, mud-dominated interior basin, this one circled in red, in Western Australia. Here you can see the major depositional cycles during time in this shallow interior sea. Now in these sand-dominated sections, it's easy to observe finer levels of depositional cyclicity. It's easy to distinguish the parasequences, for example, but not so in the thick mudstone units like the Kwinana Shale here. A cluster analysis of foraminiferal assemblages collected at 1 metre intervals in the Kwinana Shale suggests 10 foraminiferal biofacies. Their distribution is plotted against the lithostratigraphic log. You can see frequent changes in biofacies that here are related to a paleobathymetric model. An overall progradation or shallowing upward trend is suggested by the biofacies distributions. This fine cyclicity could not be determined just from lithofacies changes, and also not from carbonate chemostratigraphy, because in these mudstone units, there's a lack of consistent carbonate in most of the section. This is a stratigraphic borehole section from the Western Australian Cretaceous. Mudstone facies down here, the Gill siltstone, and then chalk facies in the Toolonga calculutite and Corridron calcarenite. The section is from here. Again, foraminiferal assemblages were documented every 1 metre, and cluster analyses of the benthic assemblages were done separately for each formation because of different species present. And the benthic biofacies plotted as colours on the log. Now the colours, the red colours here don't correspond to the biofacies here because there are separate cluster analyses done, different species present. Also, the percentage of planktonic forams in the total assemblage, there's none down here, but here there are, in the total assemblage was plotted. You can see frequent change, pointing to changes in both bottom and surface water conditions. We are still working on what these changes mean, including work with some chemostratigraphic parameters. But the fine, complex depositional cyclicity determined here could not be established just from lithostratigraphy. So in this last section of the talk, I want to show how powerful foraminiferal studies are in recognizing tectonostratigraphic units and reconstructing tectonic settings in young orogenic belts that are largely marine in character. The points made over here are all essential components of this work, and they're all derived from foraminiferal studies. The results of the foraminiferal studies should not just be service data supplied to structural geologists, who often consider themselves the most important of the geologists dealing with tectonic histories. I've been with such geologists in New Guinea and Timor orogenic belts and watched them in the field running up to outcrop and making structural measurements without determining anything meaningful about the rocks other than that they are limestone, sandstone, etc. Many of these people, the ones that I know, lack the skills to gather this sort of information. Some of them have made huge mistakes in Timor, both in age identifications and in tectonic affinities. We have revolutionized our understanding of the complex geology of Timor by using very simple techniques. And note that foraminiferal research is just as much a field science as structural geology. Don't spend all your life in a lab. In the field, we use acetate peels of the limestone samples from the abundant outcrops to differentiate these in terms of age and facets. And here in the field, Jose Nano is making an acetate peel. He's just peeled it off the rock. Very easy. It only takes a couple of minutes to make after you've etched the rock. So you have to differentiate each of the outcrops because of the pervasive faulting. And you have to even within one outcrop get ages for separate fault-bound components of the outcrop. We also disaggregate over here friable mudstone and use the foraminiferal assemblages found in the washed sand-sized residues also for age and facies determinations. We use a cement cutting saw in the field. In more detailed studies back in the lab, we can make serial acetate peels, for example, to distinguish these tiny planktonic forams present in limestone, so we can determine a more precise age. This is one specimen that has been identified in a series of acetate peels through the limestone. And it's by here you could not make an identification, but when you've got the whole series, you can make a very good identification and get a precise age. So that applies to all of these. This shows the geological position of the Timor orogenic belt in the collision zone between the Australian continent, which is moving north at around 7 centimetres per year, and Sundaland, part of Southeast Asia, part of Eurasia, which is moving to the east, roughly to the east at 3.5 centimetres per year. And Timor sits right in the collision zone. In Timor, this mainly limestone massif is a slab derived from Sundaland that was emplaced over Australian continental crust during the first phase of collision in the late Miocene. And these limestone beds are of the Australian continental crust. in contrast to these, that have been folded and faulted in the collision zone. This is our latest tectonostratigraphic classification of rock units in the Timor origin. We have clustered mappable rock units into what we call tectonostratigraphic associations. And there are 4 of these, 3 post-collision and 1, 3 pre-collision and 1 post-collision. The collision, of course, is still going on, so these are synorogenic association. Each association relates to a tectonic phase in the geological history of this region. Each association includes rock units that occur together in outcrop in separate mappable areas. For example, the overthrust terrain association, similar rock units are clustered together in each of these reddish, red-brown coloured areas. The youngest units in each association contain conglomerates, debris slides, that include clasts of the older units in the association, but not from other associations. These stitch the association together. In Timor, the ages of most of the sedimentary units, which are almost entirely marine, are based mainly on Foraminifera, with some input from conodonts and palmorphs in the Permian and Triassic. Here is a simpler diagram that shows the relationship of the tectonostratigraphic associations to the geological development of the Timor region. We have the Eastcon 1 and Interior Rift Association that extends right down to sedimentary basins in Western Australia, with interior in Eastcon 1, covering the Carboniferous through to the mid-Jurassic. Then we have the Timor-Scott Plateau Association after the Indian Ocean developed. And we find a succession in Timor like on present-day Scott Plateau, with Timor, with the Eastcon 1 and Interior Rift Association strata overlain after breakup by a pelagic cover, carbonate pelagic cover. And then over here we have the Overthrust Terrain Association, which was derived from a block rifted from the southern margin of Sunderland or southeast Sulawesi through the opening of the Banda Sea. And collisions started, I'll talk about this in a moment, but this, above the first phase of collision, are synorogenic cover deposits. Now, most limestone units forming peaks or fatus, these peaks or fatus in Timor-Leste, were mapped until recently as Lower Miocene Cablac limestone. They're the carbonate platform facets. We realised when we started our work 20 years ago in Timor that there was something very wrong with this. And now we've sorted out all the units. We've got Permian units and latest Khazalian. Here, fusulinid limestones, coral tubiphyte reef, which incidentally is part of what we think was a global warm spike. And it occurred up here in the Eastcon 1 and Rift. Further south, it led to the melting of the Pennsylvanian ice sheets that covered the Eastcon 1 and lowlands. In the Triassic, this is a slab of Bandera group, late Triassic carbonate platform limestone. These were originally put as Lower Miocene, containing the same sort of foraminiferal assemblages, lutenids and geostemenids, that occur in the Alps in Europe. The same with the Jurassic, but in another overthrust terrain association, nothing to do with the Eastcon 1 and Interior Rift Association. So these early Jurassic units, carbonate platform deposits, which again form fatus, these limestone peaks. And here, Middle Eocene, carbonate platform limestones, and our late Oligocene, earliest Miocene ones. So we have been able to reassign ages to these and place them into different tectonostratigraphic associations, essentially revising the tectonic history of Timor. Now, I mentioned earlier the importance of conglomerates in the succession, particularly in the youngest units. And Jose Nano, one of my colleagues from IPG in Timor-Leste, found a very thin, and over here, a very thin conglomerate in this mudstone succession. The conglomerate represents a debris slide of clast derived from the hinterland. It's really the edge of a big turbidite fan. In the youngest, and this is in the youngest mudstone, the Atulari mudstone of the Overthrust Terrain Association. The mudstone is dated by planktic forams, here late Oligocene in the area around the conglomerate, and higher up as earliest Miocene. So they put ages on this. And also with water depths that range at the base from the middle bathyal zone to up higher, the upper bathyal zone. Now, if we look at the different class from the conglomerates, we can differentiate through different ages the units that were supplying material to this debris slide. And these include early Jurassic carbonate platform facies with Siphovolcaniferal forams and other forams from the early Jurassic. Late Cretaceous pelagites with the planktic foram Globotruncana, and then Middle Eocene pelagites with Acarinina and Morrisavella, other diagnostic Middle Eocene types. And then a coral reef carbonate platform limestone from the late Oligocene with Lepidocyclina spiroclypeus, larger benthic forams, as well as others here. So we've been able to differentiate the units that occur in the hinterland and put that on a model like this. Here is where in the deep water basin, the Atulari mudstone was deposited. We would say 300 to 600 metres water depth in the latest Oligocene, earliest Miocene. We know that there was a shelf nearby because there were coral algal reefs and larger forams associated with them. And some of those are not incorporated in matrix. So they've come down the slope into the deeper basin. And then in the hinterland, we get a number of different units. And we recognize these in the Overthrust Terrain Association in the region where the Atulari mudstone occurs. We can see the early Jurassic carbonate platform facies, and there's a deep water siliceous argillite. There's a paper in press, a paper being reviewed on this at the moment. But these are intensely deformed with tight folding. They were in the hinterland. Then the late Cretaceous to late Oligocene carbonate pelagites, which are moderately deformed in the Maccabean overthrust terrain association block. And then volcanics of the Eocene and the Oligocene. We know from the presence of late Oligocene carbonate pelagites here that there must have been rapid contemporaneous uplift of this hinterland in the late Oligocene. And this would have allowed massive debris slides, massive amount of material to be carried through erosion, then by river transport down to the edge of the shelf, through the reef, and channels in the reef, and then down slope to this deep water basin. These conglomerates here stitch together this terrain. And the components, Otter 1, Otter 2, Otter 3, we can map out in our overthrust terrain association. Then we know this, and this came from the edge of Sunderland. And there was rifting with the opening of the Band de Sea here, probably in the Pleistocene. The or Pliocene, at the time of collision, late Miocene. The overthrust terrain association was placed on Timor in a high structural position. And we know that from the preservation of the foramens in these rocks. They haven't gone any significant tectonic loading or thermal maturity. Let's look finally at the timing of initial collision between the Australian continent and the block rifted from Sunderland. And we've recognised 4 phases of collision, but I've only got time to deal with the first and second phase. The first phase was the collision, and there was a hiatus in deposition. Now we can date the collision between the Australian continent and Sunderland from planktonic foraminiferal assemblages in the youngest pre-collision limestone. And this is now a cemented and deformed carbonate pelagite with these forms seen in acetate peel. You can see Orbulina universa. You can see limbata, Globerutalia limbata. Nepenthes, they're Globergerina nepenthes, which has been renamed. The genus has been renamed now. So this is the youngest of the pre-collision beds, and it has a N15 planktic foramassemblage. And then the dates are up here. And then we have the oldest of the post-collision, the oldest synorogenic rock unit, which is a very similar facies to this, but has not been cemented. It is standing as if it's just been lifted out of the water. And the oldest part of it is N18 in the latest middle Miocene. This is the earliest part of the middle Miocene. So we can say that the start of collision was about 8 plus or minus 2 MA. Previously, it was thought to be around 2.4 MA. So you can see the great difference. During the second phase of collision, from 5.5 MA to 3.3 MA, when subduction jammed, and there was a tectonically quiet period with pelagic deposition over most of Timor, slow emergence of the tectonic pile took place. Based on occurrences of benthic forams, which you see here, with known modern bathymetric ranges, we have been able to draw bathymetric contours on the emerging Timor origin to show this. And we use, of course, planktic foram zones to get the ages here. So you can see how powerful foram studies are in sorting out the ages and facies, not only of the rock units present in the orogenic belt, but also the timing of phases in the collision history and how bathymetry across the orogenic pile changed through time as the island denuded above sea level. No other studies could accomplish all of this. And in these studies, not too many strikes or dips or fold axes were measured. In a tectonic melange, measuring these, I think, is a little pointless. So these are the areas of foraminiferal research that I would like to pursue if I could live another 50 years. I am now over 70 years old, but I'm still optimistic. Of course, in future studies, I would be able to use many of the new technologies that are now becoming available to most researchers. And we have started to use some of these, I'll put them in blue down here, in our work at UWA. And of course, the key to all this future work would be collaboration and integration with a broad suite of other specialist studies devoted to tackling particular geological, biological, and environmental questions. So I think that's the future. Thanks.
Well, thanks, David. A very nice talk with many nice photos. That's quite a lot of work on the classification and the taxonomy and its application to the tectonic stratigraphy. So I think we have many questions to ask.
Righto, fire away.
Yeah, who will ask the first question? Any questions? Well, David, I have one question. So for the age of the start, for the initial collision, so you get the age of 8 million years old, right?
The initial collision.
Yep. The age is 8 million years old, right?
Yeah, it's. Let's go back.
How did you?
Can I go back?
Is it from the uranium light of
from the age of the planktic foraminiferal zone? You've got to remember that the neogene is subdivided by very, very finely by planktic foraminiferal zones that are linked to magnetostratigraphy in the ocean drilling program holes. And so these provide a rigorous framework in which to put your age determinations. In these carbonate rocks, you have no zircons, no minerals that you really could use uranium series dating or other methods of dating in these young rocks. But the forams, the planktic forams, provide a very fine scale in which to do this.
So there is no volcanic activities in the study area, right?
There's no volcanic. There was volcanic activity in the overthrust terrain, but not on the Australian margin.
Oh, okay.
There is no volcanic activity on the Australian continental margin.
Okay.
But in Sunderland, of course, there was, and that's why we get Eocene to later Oligocene to early Miocene volcanic units in the overthrust terrain association.
See, okay. Well, so any other questions? Well, so it's a better time to have a face-to-face interaction with David. So
someone I know.
yeah, someone might know David before. So please open your camera and have a face-to-face interaction.
Hello.
Yeah, it's quite a long time.
I don't know. Shall I stop sharing? Yeah.
Okay. It's Yukun from Nanjing University.
Yeah.
Yes.
How are you?
It's quite a long time. Yeah, fine, very fine. Yeah. Yeah, you mentioned that you had a COVID, but you seems very well.
Yes. Yeah.
Very good.
I've had all my vaccinations. I've had 3 vaccinations prior to having COVID. So I survived, but I don't think I would have survived without those vaccinations.
Okay,
So,
yeah. Okay, let me just go back to the lecture. Very nice work, and I'm quite agree with you on the for the Paleozoic Foraminifera. It's difficult to really examine them or assign them to genus or to even to species, only according to the certain slice of a certain section for those very complicated with all very complete structures. I noticed that in the you have selected some whole piece of the Trochospiral Foraminifera from the early Paleozoic.
No, from the early Permian.
Permian, yes. So could you please tell us some stories about how could you get those, or are those from the limestone or from the clastics?
They're from, I think that my student, Clement Tremlin, he's working on these with me. I wonder if he is in the audience. He was wanting to log on. Right, Clement, you can say how we extracted those. And,
let me just. All right. Hello, everyone. So when we went to Geraldton, so as David said, there's a mud facies, but in spite of looking at the shell, we looked at the nodules. So the compacted mud forming nodules, and so how you basically dissolve the rock, you put in HCl, a very dilute acid, so you will collect the residue. So I think after washing, you get those sediments right.
Okay, I can understand that. So yeah, it's like from the kind of probably will some calcite, but mainly the clastics from the clastic fishes.
These can I butt in? These are from carbonate nodules, what are called carbonate nodules. They're early diagenetic nodules. Now there are, and this is through a process of methanogenesis, and that you've got bacteria and archaea which oxidize methane and produce alkalinity in the seawater or in the interstitial water in the mud just below the surface. And this promotes the precipitation of carbonate cement in these nodules. And so it's very early in diagenesis in the seafloor, highly organic rich seafloor muds. And so they preserve these agglutinated forams, organic cemented siliceous agglutinated forams. They preserve them without squashing them. You know, things like Trochamina inflata, if you've ever collected it from marshes, you'll find even without burial, if it's dried out at all when the tide goes out, the walls will collapse because they're organic. They're partly organic walls. So they'll deflate. And in ordinary shale, you get very poorly preserved organic cemented agglutinated forams. So, but in these nodules, we've got fabulous assemblages. And not only of the Trochamina that Clement is studying, but other genera. And when we look at these, we're finding it very difficult to see where these genera originated from. You know, in the Phylogenetics, there are no trochospiral forms in the endothyroids, or you have to go right up to the Triassic, in the duostomenids, to get the first trochospiral forms. And here we've got perfect trochospiral forms. Now, Cushman and others described these from Pennsylvanian shales and Mississippian shales in the US, but they're usually squashed. But by looking at these nodules, and of course, we have just published a big paper in PaleoCubed on methane seep deposits in the Permian. The first, Jorg Pickman, who was a reviewer of the paper and is working with us now, he is a well-known expert on methane seeps. He says that these are the only genuine Permian methane seeps that have been recorded deposits, and they're nodular limestones. So we've also got getting forams out of those. But they have a different assemblage in the shale to these ones. They form from the action of methane, of course, but they give a carbon-13, delta carbon-13 signature that's very much in the negative to the ones in the normal early diagenetic carbonate nodules. So I would urge you shouldn't just dismiss carbonate mudstone successions in the Paleozoic, in the Carboniferous and Permian. Too many people, and I've seen it in some recent papers and they're usually the people who study carbonate platform faunas. And there was ridiculous comments like the carbonate platform foraminiferal faunas are really the only foraminifera that lived during the late Paleozoic. They're very poorly, represented elsewhere. And yet in the mud, and that applies also, they've said this for the Triassic, that also, they haven't looked in the mud facies. There is a abundant, prolific fauna with species diversity above that in most of the adjacent carbonate platform deposits. But they never look. They don't know how to recover the forams from the mudstone facies. And
yes,
this has been a problem. And of course, a very noted Paleozoic worker in France, late Paleozoic worker in France, has said that all these organic cemented agglutinated types, which are siliceous because they agglutinate quartz and other silicate minerals, that these are just recrystallized calcareous foraminifera. So, and that's not true in these deposits. And it's not true in modern estuaries. That's why I'm urging people, every foram worker should go to the seaside and should look at estuaries, go and dredge some mud, wade into the water and take some mud and look at it. And then go into marshes and take some of the marsh vegetation, and particularly around the roots and the sediment. You've got beautiful subtropical, tropical beaches in China. You must have beautiful seagrass meadows. Go down and look at the forams on the seagrass and see how they live. Many foram workers don't do that. Many foram workers don't actually move from their particular stratigraphic horizon. It's as if they're trapped in a narrow time span.
Yes, thank you for your suggestions. Definitely important to say the modern foram and the if we can see more forums in all the ages to see the diversity and all the possibilities, to see the whole picture. Yeah. Thank you for that.
Right.
Okay, David, there is another question from the chatting box. So I have some questions about the morphological study of forams, that is the coiling direction. What decides the coiling direction? Genome, temperature, or salinity, or other environmental factors? Whether the relation... Sorry, yeah, okay.
Oh, go on if there's more to the question.
Okay, so whether the relationship between the coiling direction and the environmental factors are reliable. Can these relationships were used in Paleoceanic reconstruction? Whether the study, well, it's quite a lot of questions, but whether the study of the coiling direction of forams is of scientific value. I look forward to hearing your opinions. Yeah, this is from Shuai Dong. Yeah.
Righto. Now it depends on the group of Foraminifera. And in most Foraminifera, trochospiral Foraminifera we're talking about, the coiling direction is random. There's no environmental factors that I can see. But there are some planktonic Foraminifera, particularly in the Neogene, and particularly in the late Miocene. There's a form, Neogloboquadrina, and I'm trying to think of the species name. It's just escaped me at the moment. But the change from sinistral to dextral in that form is very important stratigraphic horizon. So in some of them, it is important. But the cause, I haven't followed that literature for quite some time, but the cause, I guess, is environmental. And I don't know whether it's synchronous. I don't think it's synchronous between ocean water masses. So in the Atlantic and in the Pacific, it might not be synchronous. I'm not too sure. But there are these definite changes in coiling direction. But in the majority of benthic forams, I don't think that it is important. And they coil whichever way they like.
Okay. We'll see. Any other questions? Well, I have another question, David. So what is the age of the oldest forams? Is it Carboniferous or or it is maybe much older?
No, they're probably in the Neoproterozoic. You should
really?
be finding them.
Oh, okay. So,
They are. They're definitely in the Cambrian, but not the one that the late David Scott described from the Cambrian. He described Trochamina. These are just squashed some aggregation of grains. So there, I dismissed those. So they're simple, very simple tubular or spherical forms that may be, it's very difficult to say that those types are foraminifera. The first multi-chambered foram, and it's one in which the chambers are added one above the other in a straight line, rectilinear, is Rheofax that occurs in the Ordovician, described from the Ordovician. And in fact, the same Rheofax, the same type of Rheofax occurs in all our deposits in Western Australia from the Permian right through to the Cretaceous. And we get similar ones in our modern, but in deeper water, our modern assemblages. So Rheofax is the root stock, I think, of all multi-serial, multi-chambered foraminifera. But then you come on, it's interesting that the first planispiral forams are probably within the, well, almost planispiral. See, a planispiral coiling mode can be very irregular. And that's why I think the endothyroids have gone into a streptospiral mode, many of them, because it's very easy from a planispiral mode of coiling, the irregularities to give a streptospiral coiling. But the first trochospiral ones are those ones in the Mississippian of the southern US that I think Cushman and perhaps Walters described first, but Conkin and Conkin renamed the species Trochamina howii. And then you see those also very poorly preserved, but enough to say that they're trochospiral. We can't see clearly the apertures in them. And this is a factor, the type of aperture is important for classification of these things. You have to, and in the Pennsylvanian, there are forms, there are a couple of species, but the apertures are a bit obscure. But in the Pennsylvanian, there are nodules in the shales in the US, and they should be looked at. They would solve a lot of problems. And then you come up to Australia has got perhaps the best interior basin successions of mud facies. Very shallow water without very much burial. So we get beautiful, both Milliola assemblage, calcitonellids, etc., Henigordius, etc., and beautiful nodosarian assemblages in these rocks that haven't been buried at any depth. And so we can see in the assemblages that Clement is looking at of Trochamina, we can clearly see the aperture. And in fact, Sylvain Lagarde, who is a friend and who worked in Singapore but is now back in France, he commented when we sent him images of those of how advanced the aperture was in those early Permian types. And so there's a question mark of where those the pathway from Rheophax to those Trochospiral Trachamina. Now, very few studies have been made of mud facies, and it's very difficult. We've got good Carboniferous up north, but it's been buried 2,000, 3,000 meters, 1,000, 2,000, 3,000 meters. So the preservation won't be so good. The carbonate, shallow water carbonates are very well preserved, and I'm looking at the Mississippian there. But we've got a gap because of the Gondwana ice sheets in the Pennsylvania. But I don't know what's in China, whether you've got good Carboniferous interior basin mud successions.
Well, there is indeed some Permian to Triassic sedimentary sections in South China that potentially contain the forams. And some people are now doing such work, I think, such as Nivison.
That's good to hear. But let's hope they're extracting the forams from the rock. Because you can only, these smaller forams, you can only really do when they're extracted from the rock. But the thing that we're now looking at is micro CT scanning and nano CT scanning of this. Of these. And some of the companies in Perth are using that on sidewalk cores and so forth. There are other techniques that will become, I'm sure, in future become available. So you don't have to disaggregate rock, but you could image in 3 dimensions what's in the rock.
Good. So do we have any other questions?
Hey, David. Hey, can you hear me?
Yes.
Hey, David. My name is Xing Hai. Come from Chinese Academy of Science. I have one question. You know, for benthic foraminifera includes one informal group. It's called larger benthic foraminifera. They usually contain symbiont algae and live in very shallow, warm marine environment. Like the picture, I think you showed me in the first of your PowerPoint. I think this maybe also belong to the larger benthic foraminifera because it contains some algae. So my question is, do larger benthic foraminifera, can larger benthic foraminifera survive in suboxic or anoxic water column?
I would say probably not. Now, the foram I showed you was Peneroplus. And what I'll correct you, it's not only larger benthic foraminifera that have symbionts. There are smaller benthic foraminifera and planktic foraminifera that have symbionts. So it's not exclusive. We talk about larger benthic forams, and Peneroplus that I showed you at the start, it's got a fairly simple spiral chamber arrangement. And although Peneroplid limestones first appear in the Cretaceous, late Cretaceous, for example, in southern Europe, in Spain, for example, and they're regarded as larger benthic forams. I think, when I think of larger benthic forams, I think of things like the discocyclinids and the astrocyclinids, the alveolar annelids, and very complex chamber arrangements, the nummulites, and in the late Oligocene, early Miocene, the lepidocyclines and Spiroclypeus and Cycloclypeus that can reach large size but have very complex chamber arrangements that you have to look at in thin section. Just like the Fusculinids are probably had symbionts in the Carboniferous and Permian. And I suspect that the involutinids, things like Allotortus and all those forms in the Triassic probably had symbionts. Their shells, their test, seem to be glass houses. And that's the other factor of these forms with symbionts. Their shells are glass houses, and they farm the unicellular algae, whether they're diatoms or whether they're these purple-coloured rhodophytes, unicellular algae, in Paneropolis and Marginoptera and Amphisaurus, etc. in the modern. So you've got to, and as to many of these, so they need light. And in carbonate platforms, the best substrates for life, of course, are living as epiphytes on seagrass, in these extensive seagrass meadows that go down to 30 or 40 meters water depth. Beyond that, you get very large potato chip, like very thin, larger forams. We can tell that in our Fassie's work on discocyclinids and astrocyclinids in the Eocene, and we can see it in Cycloclypeus. As you go deeper and start to get in the Fassie's, more planktic forams coming in, you'll see that these larger forams they thin out and become very large in size to capture light. Now, you know, if they're living above the substrate on seagrass, the sand under the seagrass, of course, includes a low oxygen environment. You often get in the sediment under seagrass pyrite, iron sulfide, forming in the sediment and turning the carbonate grains grey in colour. We've got whole beaches. I did work on this for a city council here in one of the coastal towns here, and they were having concerns that their beach was turning grey. And what they didn't realise was that in winter storms, the seagrass is ripped up and the sediment under the seagrass is transported to the beach. And when you look at it, a lot of the carbonate grains there, whether they be forams or mollusk shells or whatever, or little limestone fragments from our coastal limestone, they're full of very tiny aggregates of pyrite. So you get these microenvironments in the sediment under the seagrass. So I think it's very important to work out what's going on in the modern environments. So the other thing, not only do foram workers and other paleontologists who are doing Fassie's analysis have to know the microenvironments in which the forams and other organisms live, but they also have to know the sedimentary environments and put everything in place. So that, and really there has to be an integration between the ecologist in the biological side and the foraminiferologist on the geological side to understand these.
Okay, David, thank you very much for your answer. So I know that the symbiont algae foraminifera, they need light, but I also know most of them, maybe all, live need oxygen, but I'm not very sure if oxygen is extremely necessary for the larger foraminifera. So you tell it's yes.
Well,
yeah.
the larger foraminifera get additional oxygen while they can produce such large calcium carbonate shells, whether they be high magnesium calcite or calcite shells, is that they acquire from the unicellular algae the additional oxygen that the algae, like all photosynthetic organisms, expire. And then the algae also are giving them their nutrients, their waste products as nutrients. So it's like a farm, having a farm in a glass house. And producing, as you see with Paneropolis, there are hundreds, there's perhaps thousands of embryos produced. And each of those embryos has taken, I haven't counted them up, but maybe 100 symbionts, unicellular algal symbionts, an immense number of symbionts in that Paneropolis. So it, and so they're getting, the foram is getting additional oxygen and it's getting additional nutrients from the symbionts. It needs light. Now, there are different, and really in methane seeps, it's an interesting case because there are methane oxidizing and symbionts there. And it'd be interesting, and some work has been done, I know it is being done, but it would be very interesting to look at the relationships in modern methane seeps of the foraminiferal assemblages. In our paper, in 2000 that came out earlier this year in PaleoCubed, we recorded the foraminifera, but they're described in the supplementary material thing.
Thanks.
So, any other questions? So, the Clement, I see you open your camera. Do you have a question? The Clement, you have a question?
I'm good. The question. No. Good.
He was answering the questions.
Okay, so, well, if there
Can I just occupy a bit of time? David, we definitely for myself, I've also did some CT scan for the Paleozoic foraminifera. It's definitely for us. It's definitely need to say that the 3D versions of the Paleozoic foraminifera, but most of the time, what we kind find is the foraminifera preserving in the limestone. So I think the much common way to study them should to do the successive peeling. I've noticed that when you do, you did some peeling in the field. So what's the reason for that? Yeah, what's the advantage to do that in the field?
You know, the geology of Timor is so complex. Hamilton, the US Geological Survey geologist in the 1990s or 1980s, he came to Timor and he said it was one of the most complex geological melanges in the whole world. And so you have to identify each small outcrop in terms of age. And they're all mainly limestones, and there's mudstones intervening. And even within one outcrop, you can recognize where there's faulting, but you have to date either side of the fault. Now we do that. We take saws, we slab the rocks in the field, and we take them back to the base camp at night, and we etch them in 2% HCl acid. And you don't have to polish them before you etch. Just the rough saw cut will be sufficient. And then you etch them in 2% HCl. And then when they're dry, they must be dry. You then, and as you see, we put them out in the garden, and so we can lay them flat. And you get a bottle, a wash bottle of acetone, and you flood it with acetone. And I buy acetate sheets from the US. There's a company called Blick that sells very cheaply big books of acetate sheets. And we put a sheet of acetate, cut the acetate sheet to the size of the rock and then put it over. And within 2 minutes, you can take off the acetate sheet. And then we've got field microscopes. We put them between two glass plates. We buy picture frames, 6x4 picture frames that you put your photos, child photos in, etc., all made in China. And we buy 2 of those and put the acetate peel between the glass sheets, just tape it down while we're looking at it under the microscope. And it's better than thin sections. A thin section, when you look at it, will have a thickness in the limestone, which can be very confusing. But an acetate peel will give just the surface, etched surface. And the cement fabrics, the wall structure of the forams come out perfectly. And those things can be made in the field. So when we go out the next day in the field, we know exactly what we've been through.
Okay.
That's why we do it. And before, most of the other people who have looked at the geology of Timor have just collected rocks and taken back to their own labs in Europe or in North America, etc., in Australia. And, months after, they try to sort out what they've seen. And they don't really know what they're looking at in the field. So I think that for foram workers, it's in those sort of terrains, it's really field work that is important.
Thank you. Impressive work.
Mm-hmm. Thank you. Yeah. Do we have other questions? Come on, everyone. So if there is no questions, I'm afraid so this is the end of the talk today. Yeah. So thanks very much, David. Yeah. Thanks very much.
Yeah, thanks very much for listening to me.
Yeah. And also thanks for everyone for your participation. Yeah. So yeah, looking forward to have collaborations with you in the future. Yeah.
Yes, well, as I said, that collaboration and integration, that's the future.
Yeah.
That's what we must do.
Yeah.
So I look forward to seeing many of the Chinese colleagues at Forams 2023. We would be there now in Perugia if it was originally scheduled, but it's been rescheduled to next year. So.
Okay. Yeah, that's
we should really talk about collaboration. Yes.
Okay, great. Okay. Yeah, hope we can meet in the project. Yeah.
Yeah.
Okay.
Righto. Yeah.
Okay.
All right.
Yeah. See you.
Bye, David. Take care. 谢谢蓝老师、吴老师。
客气。好。 再见大家。